Good afternoon, everyone. My name is Ken Buetow. I'm from Arizona State University. I'm the Director of Computational Sciences and Informatics. I'm going to be presenting today on a topic of interest to my particular group, and I hope to you all, about embracing the complexity of cancer, seeing forest and the trees as we attempt to move forward in understanding the world of cancer. Just to remind you all of the logistics, we'll be taking questions and answers at the end. Please feel free to submit questions into the question and answer box. When I complete the talk, I will address the questions that have come up through the course of the talk. I'll read the questions and provide answers, and hope that you have an interesting and enjoyable engagement. So let me get started. We're in an interesting and I think exciting transition in biomedicine into personalized, precision, genetically specified, you name lots of names for it, where we are actually changing the very fabric of medicine to being much more personalized. This change is about providing targeting the right individual, the right care, the right time, in the right place, and in many ways is a fundamental revolution as we embrace in personalized or precision medicine the unique molecular and biologic characteristics of an individual, but also it's a bit of a renaissance as we not only return to actually bringing in not only the unique molecular characteristics of an individual, but the unique clinical, personal, social, as well as experiential capacity of an individual. What's exciting about this revolution or renaissance is that, in fact, it's actually very real. One of my favorite quotes is, "The future is already here. It's just not evenly distributed." And in fact, this world of personalized precision medicine is already in front of us in many places. So from my local newspaper, I pull up advertisements from the Washington Post or other places that indicate places that are already well engaged in performing this type of precision or personalized medicine. This is from Vanderbilt University, and I'll show you work courtesy of my colleague Mia Levy at Vanderbilt and the outstanding work they're doing in My Cancer Genome. So what one can do in the cancer field today is go to My Cancer Genome, select a particular disease, in this instance melanoma, choose a particular gene of interest. If you know that you have a mutation in the BRAF gene, what is the specific mutation? Shown here is the V600E alteration, where there's a T to A change in BRAF, and now one can derive personalized precision medicine, finding out, given that my melanoma has this particular form of molecular alteration, I can, drilling down on that slide, see what types of interventions are available to me and what types of drugs might be able to be used. You can see there's BRAF inhibitors, MEK inhibitors, KIT inhibitors, with specific degrees of evidence associated with what might actually happen and what drugs I might use and what's been the outcomes associated with this. So we're actually at the very beginning of this revolution and seeing that, in fact, there's really exciting things we could do today. But one of the critical things that you might guess, given that I'm from the complex adaptive systems group, is that while this is an exciting beginning, we are just beginning to scratch the surface of what it means to have precision or personalized medicine, specifically in the cancer field, which in many ways has some of the most interesting but still challenging problems in this space. So what do I mean? So while looking at a single gene is exciting, and does provide real opportunities today, when we actually look at what we know about cancer today, it's actually pretty complicated. And in fact, way more complicated probably than looking at a single mutation in a single gene if we really want to be able to have an impact in dealing with cancer. So let me spend a minute or two walking you through, when I say it's complicated, what I mean. Well, first, cancer is a complex adaptive system. So that if there's a key take-home message I would like you to get from this, it's just exactly that. Cancer is a complex adaptive system. So let me walk through that. We know that cancer starts in a base state and accumulates alterations over time, much the same as any sort of evolutionary adaptive system, where it accumulates multiple mutations and in fact creates a new organ, or conceivably, depending on your point of view, new organism within you as an organ. So it creates a three-dimensional new collection of genetic constitution that has all the complexity of any organ or organism, but it has adapted and grown in real time. These portfolio mutations that occur over time are not uniform in every cell, as the diagram attempts to show, and in fact number in the hundreds. So there's not a single mutation that occurs in cancer, but in fact literally dozens to hundreds of mutations that occur in cancer, or alterations that occur in cancer. The next thing that's important to know in this complex adaptive system is that these alterations can occur in a variety of ways. As a consequence of the Genome Project, we spend a lot of time thinking about how we would be altering the specific constitutional DNA that's part of an individual. But we also recognize that mutations in your DNA are but one way that we can generate these alterations. One way we can generate them in addition to mutations is by changing the copy number of a gene. We can generate alterations by changing the expression of the genes, not just necessarily through mutations. We can generate alterations by epigenetic mechanisms that aren't direct nucleic acid modifications, but in fact are modifications to proteins, histones, and other components that alter how the nucleic acids are expressed or changed. We can have alterations not only in these epigenetic processes, but in also other processes that are completely outside of the expressed genome in the traditional sense of protein coding genes, but actually genes or microRNAs that actually change the properties of genes themselves. And lastly, we can have processes that change these or generate these adaptations or changes that are completely outside of the protein and or nucleic acid space in glycolysis or other types of chemistries in a cell. So now, if this isn't complicated enough for you, what we see is that the complexity of this system is manifest not only in the number of changes it can make, the three-dimensional structure and the processes it can alter, but it's also involves a number of complex biologic processes. We've known for well over a decade now that there's several key processes that are important in the development of cancer: growth factor independence, evading apoptosis, telomere maintenance, cell cycle regulation. But what is hidden by those simple soundbite class says that we have to change, evade apoptosis, or change the maintenance of telomeres is that those processes themselves are products of complex networks, interacting collections of molecules themselves. And to actually address growth factor independence, one can mutate one of many of those genes in those processes in a given individual, but another individual can still be altering those processes, but change another completely different collection of those. Or another individual can change yet another collection of those. So in fact, we can change, we not only have the ability to actually change individual genes, but if we look at it from the lens of a process, there's not a fixed set of genes that need to be changed in order to alter a particular property that's necessary for the generation of cancer. Lastly, if this isn't complicated enough, this somatic collection of things, what we realize is that this somatic changes occur in a very complex environment of genetic constitution. There are anywhere between 1 million to 10 million individual variants that exist in your genome that differ between the DNA you inherited from your mom versus the DNA you inherited from your dad. Those variations can influence a variety of endogenous processes, how you generate blood vessels, the underlying structure of the cellular matrix, how you respond, what your basic immune response is. Additionally, they can not only change those processes, but actually act at the interface to the environment itself. So they can change how we respond to chemicals we're exposed to, viruses we're exposed to, different hormonal milieus, as well as how we would actually respond to different nutritional aspects. So With all that said, I guess I would say that hopefully cancer is not only complicated, it's very complicated. So cancer is a complex adaptive system. Why does this help us? Well, if we recognize the whole is more than the sum of the parts, it allows us then to attack the system as a whole rather than addressing the individual pieces of cancer. So recognizing that cancer evolves over time, there is not one such thing as a static tumor genotype or tumor molecular profile. By embracing the fact that it adapts, it has dynamic as opposed to static behavior. By realizing that it expresses emergent properties, that in fact looking at through a completely reductionist mirror or completely reductionist lens, we will miss emergent properties. There are pieces where the whole is more than the sum of the individual parts. It requires multidimensional or transdisciplinary study. It requires there's not a single point of view that's of utility in looking at cancer, and that we need to embrace its interdependencies, that cancer itself shares and manipulates resource, and at some fundamental level, as a complex adaptive system, its key variable is information. So what I want to focus on here is those interdependencies. The strategy that my group looks at is how do we leverage and understand and use these interdependencies to better understand how cancer emerges, and hopefully then to be able to ultimately target how we will intervene and change the performance. So our fundamental view of this is this interconnected network-based world. So as I'm going to move forward, the key metaphor that I'm going to use is these networks of interconnections. So early in the cancer molecular revolution piece of personalized medicine, the somatic genetics of cancer drove the community to consideration of network models. When one started looking at genome-wide characterization, such as exome sequencing, RNA-seq, whole genome sequencing, as I mentioned a moment ago, there's literally hundreds of mutations observed. But what was confounding is while those hundreds of mutations were observed, less than 5% of them reoccurred from individual to individual to individual. So there was an observation of a very high frequency of very high frequency gene and or gene-specific mutations is very rare. It also observed that when we look between tumor to tumor, when we look at mutational signatures or expression signatures or epigenetic signatures, there's a ton of variation. It's very hard to find uniform, coherent signatures from individual to individual. And it also became clear that the multidimensional data provided apparently conflicting results. Apparently conflicting until all this data was projected onto biologic processes and biologic networks. Shown here is one of the earliest examples that emerged out of the Cancer Genome Atlas project with respect to the cancer, with respect to glioblastoma multiforme, GBMs. And what they saw is a very complex pattern of cancer at an individual gene level. So for instance, if you look at EGFR that has a very high frequency of mutation, you look at ERB2, it has mutation rates of less than 10%. And you look at RAS, mutations in less than 2%. But what we saw is when we project all these various types of alterations onto molecular networks, there suddenly emerged a coherence. The chaos evaporates, and you can see that there's actually systematic patterns and that different cancers in different individuals are targeting a common underlying biologic set of interconnections and then manipulating those particular processes. So these accumulations of these very diverse patterns of alterations are actually geared at disrupting these key underlying networks. And when one projects onto these networks, you can see that, for instance, the RAS pathway, rather than having single-digit gene-specific mutations, when we look at the RAS pathway, 88% of the tumors disrupt the RAS pathway. 87% disrupt the p53 signaling pathway. 78% disrupt RB signaling. So we start to get coherence by projecting the data into these network models. So my group has been particularly interested, given the session that I'm in looking at the quantitative approaches by which we can find these processes so that we can objectively find these processes rather than just descriptively find them. Some of the earliest work in my group was done by my postdoc, Saul F. Roney, who's now at Bar-Ilan University. He developed a tool that's now instantiated in the program Pathologist that has later taken gravity or taken form in a later publication by the group from the UCSC in the Paradigm work. But Saul published this work, as you can see, in 2007 as the sort of first way of computationally approaching the state of networks by looking at their interactions. So what do I mean by this, and how is this different than basic signature analysis? Well, first, let's look at how the algorithms and the analytics actually work. So here's a cartoonish network. We can see it's composed of 3 interaction nodes and a total of 1, 2, 3, 4, 5 different genes, or as well as this complex DE. So 6 different interacting inputs. And what we do in this is then paint the data for any given observation. On this, you can see the A, B, C, D, E, F, G at the bottom. We can press their state at the bottom, and then project that data onto the network diagram itself, and then compute at each of the nodes as to whether, in fact, once we've laid the individual gene state, as to whether we're getting coherence or activation at each individual node. So what does this look like in practice? So let me walk through a specific example. So let's start by looking at node 1 and the gene states for A, B, and C. We can see A is present, B is absent, and C is present. From the little cartoonish things, the arrowhead means that would activate the interaction, and B, if it was present, would suppress the particular interaction that would actually prevent us from seeing C. So if we actually look at the state of A, the state of B, and what is the product state of C, we can see that, in fact, that particular interaction is consistent and active. So interaction A is active and consistent. Again, integrating the information from the states of gene A, gene B, and gene C. Let's try it again on interaction 2. So in interaction 2, we now lay on the state for B. A was still there, and now we look and see what the state of D is. Okay? So now when we look at interaction 2, we can see that both A and B should be present in order to have D. In fact, what we see is that's not correct. So what we can say from this analysis is that that interaction should not be active and is inconsistent. Lastly, we can look at interaction 3. And in interaction 3, we have the complex A and B are both present. We have D is present, and we have F is absent. And for interaction A to be active, it needs both the presence of E and the absence of D. And what we can see then is, given that F is absent, that we can say that, in fact, interaction 3 is inactive but consistent. Okay, so we can work that through all of these 23,000 genes, through all the existing network models we have, and then calculate pathway signatures as opposed to gene signatures. And what we can see is we get very different results by using gene-based signatures than using pathway signatures. So in some ways we can reduce dimensionality or complexity. One of the vexing problems of analyzing genome-wide pools of data is the numeracy of the dimensionality. And by looking at pathways, we collapse that information and actually could do it all the way down to looking at the overall state of the collection of interactions in that particular pathway. So we could either score this pathway as having 3 units or even all the way down to 1. We can also see that it can reduce differences. So we can see that if we looked at the gene signatures for this pathway, as opposed to the pathway consistency, that there are 4 differences. So it looks like there's a lot of difference between individuals. But if we look at the pathway level, we can see that, in fact, all of the individuals have a consistent pattern for their pathway. So there's no reason to think that this pathway may be then have a differential activity in whatever phenotype, cancer in particular, that we're looking at. Lastly, we can see that it can amplify differences. So by looking at the network interactions and the gene states themselves, we can see that in the first example of using gene signatures, there's only really one difference between the two sets of signatures. But if we look at pathway consistency, scoring at the intersections, we can see that in fact this amplifies the differences because of changing a single gene can have a radical effect on the form of the network. Okay? So what I want to do is actually move from the somatic piece to a further work that we've been interested in is how do we actually look at constitutional variation. And this was work done by, again, a postdoc in my lab, Rosemary Braun, who actually allowed us to take the emerging very large collections of GWAS data that exist and say, how do the patterns of that change if we use pathways as our level of analysis rather than genes. As those of you all familiar with looking at GWAS class studies, there's been a vexing complexity in that we find very low odds ratios, very low effects associated with individual genes when we look at GWAS studies, and oftentimes difficulty even in large-scale studies of reproducing the findings from study to study to study, and even when they are reproducible, the effects are very small. Our viewpoint, and I think I'll attempt to demonstrate by the reanalysis of a historic data set that Rosemary included in the paper that I showed here, that in fact, by looking through the lens of networks, we can get interesting additional results that I think are both interesting and provocative. So we looked for this particular example, we used, and what covered in Rosemary's paper is the CGEMS breast cancer study population, the cancer genomic markers of susceptibility, originally published in 2007, a very large case control study that had a genome-wide collection of SNPs characterized on the Illumina pathways, and we then looked at the molecular state of those pathways by looking at the variation and looking for differences in the pattern of variation in these particular genes that are part of these pathways. Different from gene set enrichment or other types of analysis in the sense that individual SNPs don't have to be specifically significant in order to influence the molecular state of the pathway. And in fact, this type of analysis aggregates and captures the emergent properties by doing distance calculations, looking at all of the individual SNPs in a pathway simultaneously, so that one doesn't have to first have universal significance of a given SNP and can see the combinatorial effects of multiple SNPs being considered at once. So shown here is actually a pathway of interest that won't be particularly clear. Our group has the key finding historically in the CGEM study was the greatest locus of significance was FGFR2 by itself had an odds ratio of 1.3, a very highly significant odds ratio. Shown in this diagram here in the red circle is where that FGF2 sits in a much more complicated network that we chose to analyze. And what you can see, not surprisingly, even though we're looking at breast cancer, we can see that pathway names actually are just like gene names, not always particularly descriptive of what the pathway is doing. But you can see that FGFR2 with a different gene nomenclature here is a key intersection point in driving multiple networks associated with apoptosis, cell cycle, p53 signaling, and other key response to androgens and other sex hormones as well. So it's at a very interesting juxtaposition. So if we actually now look at this pathway, instead of just FGFR alone, we can see analyzing this, the original result that was published in the original paper by Hunter et al., they got an odds ratio of 1.3. If we actually look at this pathway, we can see that this pathway itself has an odds ratio when we look at all the genes in it of a slightly higher value than the original odds ratio that Hunter and all observed, 1.38 as opposed to 1.3. But again, wildly significant now because of the multiple combinations and the reduction in complexity. But what becomes very interesting is that actually if we drop FGFR2 out of this, we see that the residual members of this network also contribute to the significance. So while FGFR2 FGFR2 has a very high contribution to the overall significance of this pathway, we can see that other members of this pathway equally contribute to differences between cases and controls. So showing that one of the reasons we may have difficulty in finding both higher odds ratios as well as understanding the complex heterogeneity of something like breast cancer is it's not due simply to FGFR2, but multiple alterations in a variety of members of this gene network. So shown here, I apologize, it's a little bit hard to read, but you can see FGF2, the prostate cancer network, actually isn't even the most significant network if we look at a network-based analysis of that data set. In fact, we can see that there are other, well more significant pathways that are showing differences between breast cancer cases and controls. Not surprisingly, things in purine metabolism, but interestingly to folks who are familiar with the epidemiology literature is we see calcium signaling as an important pathway modulating risk for breast cancer. And there's been a large literature that's grown up around the observation that calcium intake may be important in determining risk for breast cancer. And in fact, this may be one of those variants or network collections that I showed earlier in the slide of cancer that where we have a network mediating the interface with environmental exposures, driving individual risk. So we can actually then look to see what would happen if we started to combine pathways to the overall odds ratios of predictions of risk. So as I said before, we saw that a single gene, FGFR2, has an odds ratio of 1.3. If we actually combine the top two pathways, we see our odds ratio now goes up to 2.0. And if we actually combine multiple pathways all the way down to the bottom, we actually move now from less than a 30% increase in risk to now over 300% increase in risk by capturing the variation in a number of pathways. So we change the character of the problem and actually move it into a place by looking at these networks of having profound significance between the cases and controls, not just modest significance, and actually having things that are much more predictive and or diagnostic of what can occur. And I'll point to that 3.38 as what you can get from this particular single data set with whopping high significance, 10 to the minus 95 in terms of its significance. Compare this to the recent publication that came out of, that received a lot of coverage of combinatorial studies, and I'll point you to the breast cancer line, that you can see even with these combinations of multiple studies, the highest odds ratio that one gets considering the highest 5%, so this is already truncating, is 2.4% compared to 3.3% that we got before, looking at this broader population. So one might argue that the use of these networks provides a very fundamentally different view of what the problem will look like and may help us better understand what's occurring in terms of broader genetic susceptibility. So I want to transition here for a second, because one of the things that I think it's important for us to realize is that if we're going to embrace this large-scale collection of problems, embrace the networkedness of cancer as a disease, and all of this multidimensional data, what we actually realize is that personalized medicine is a big data problem, okay? So we've heard a lot about big data. You can't actually open a newspaper nowadays without hearing people talk about analytics and data and how big data is going to revolutionize almost everything, in particular biomedicine. I'm going to spend the next portion of my presentation here talking through how through the embracing of big data and treating personalized medicine as a big data problem, we may be able to help not only get coherence through understanding the complex adaptive systems nature of cancer, but we may be able to ask and answer very complicated questions that are still largely vexing us when we look at data not in a big data context. So let's start with, what is big data? It's probably one of the most heavily overused words right now. So big data is minimally, not surprisingly, big data. So one of the key dimensions of big data is size. But volume isn't the only thing that characterizes big data. Big data is also characterized by variety. Variety meaning many different sources, much heterogeneity associated with cancer. And then over and above that is velocity, where we need to actually process the data very, very quickly and then be able to move that data and utilize that data not in days, weeks to months, but hopefully as close to real time as is possible. So unequivocally, when we actually look in the personalized medicine space, whole genome analysis, all the work that I've just mentioned, is a critical dimension of defining cancer and personalized medicine as a big data problem. Shown on this slide, you can see these are numbers for a single sample. The raw data that comes off of next generation sequencing generates 220 gigabytes per sample. Even as we start to process and reduce the data, we're still into hundreds of gigabytes in an assembly, and even the variant identification resources are measured in hundreds of megabytes per sample. It's also big data when we talk about the velocity dimension. To get real-time or near real-time processing of raw data to assemblies and assemblies to variants, you can see it takes significant computer resources, dozens of computers. When I say 24 cores, that's literally 24 separate processors and computers working 72 hours to work on a particular problem. So you can see getting to velocity is a very big challenge in the big data space. But let's be frank, genome data is but one dimension of the big data necessary to create personalized or precision medicine. It is in some sense dwarfed by when we bring in data from the phenome. So as a classical biologist and geneticist, the phenome is all of the clinical description of an individual, its phenotype. But over and above the phenotype of an individual, what dwarfs even the phenotype characterization is the exposome, all a lifetime's worth of characterization of all of the external factors that touch and conceivably modify an organism. All of these are tremendously large sets of data. So let me start with the phenome data. The phenome data is huge. It's got diverse types. It's got clinical observations, clinical laboratories, imaging, registries, biospecimens, and reference, knowledge-based sources. More vexing, and again, back to the both velocity and variety, it's distributed in research settings, care delivery settings, and even the care delivery setting isn't uniform. It's got hospitals, individual clinical practices, laboratories. It's present in registries. It's present in industries, and actually consumers contain and hold key pieces of this information. Focusing on just one type of this phenome data that in and of itself dwarfs the genomic data is imaging data. Shown here is a cartoon of whole body imaging associated with drug delivery that's going to be increasingly common as we have non-invasive ways to measure our interventions. Each of these individual images is as large or larger than the genome spaces that I just mentioned a moment ago. However, these are but the tip of the iceberg, and when we talk about both the phenome and exposome, the true explosion of the data, the next big data challenge, is going to be in real-time consumer data. This is data that may be monitoring our internal clocks, such as our cardiovascular disease or diabetes, or external or other related monitors, monitoring our food intakes, our fitness, or other things. And just as of this year, it's expected that there are going to be about 2 million of these devices generating continuous streams of data. So this is pretty overwhelming. How are we going to deal with this? Well, again, similar to what I said before, what's exciting about this kind of stuff is the future is already here, and perhaps borrowing a line from the, or paraphrasing a line from the movie The Graduate, I have But one word for you, Benjamin. How are we going to deal with this big data challenge? Google, or at least Google-like approaches. Groups like Google actually have mastered a whole new technologic and/or data infrastructure that blurs computational capacity with large data resources and novel algorithms that allow us to find patterns in data in near real time. So when we talk about large volumes with variety, it's hard to think of more variety of data than the internet, and velocity. Google processes results in real time. So what do I mean by that? It's actually doing analysis real time. Well, almost everyone is familiar that as you now start to type into a Google search window, it starts to guess what you want. It's doing that by leveraging real-time analytics that it has performed by analyzing both millions to billions of web users like yourself as to what types of words do they put in, and creating complex interaction maps, not unlike the maps I showed for the molecular diagrams, what actually are common patterns of use that an individual might be interested in as they are trying to do their queries, anticipating what might happen by using these complex interconnection collections of networks. Another key example of this would be Amazon, who probably in many ways is the master of this in consumer space, where I think every one of us is familiar where we go to Amazon, perhaps enter a particular search query for a book that we're interested in. And here's a query I did about looking for a book about big data in the emerging space of how we will do interpretation and analysis in big data space. But as everyone is familiar with, as soon as I do my query, Amazon immediately figures out that there might be other books that I might be interested in. And it does this, again, by using the underlying correlational network structure and big data processing through things similar to the Hadoop file systems, MapReduce prices, MapReduce analytics, and a variety of other sophisticated statistical techniques that combine and create complex network maps underneath all of these different pieces. So we're very interested in pushing this envelope in the fourth paradigm science, data science-driven work, where in this instance, we actually use as our experimental platforms, not actually bench-based chemistries or PCRs or molecular analysis, but actually use this explosion of data in fourth paradigm science to complement how we do both discovery and validation using data as our primary reagent, not necessarily some wet reagent. We think that it complements data generating science. Quite commonly when people talk about the current type of data, they talk about hypothesis testing. I actually think that that's unfair to data science to say that we don't do hypothesis testing. Folks that have been doing data science for literally hundreds of years, astronomy, physics, economics, don't actually go out and move the planets. So I think data science can be just as important in testing hypotheses as traditional bench-based experiments. But the key difference here is we're using data-driven science as opposed to generating data and then analyzing it. So the fourth paradigm science is really data-intensive science. It moves from the first paradigm, which was just empirical descriptive science, theoretical models in second paradigm, computational science in third, and now data-intensive science, where data generated by large-scale instruments or simulation is processed by software. The information and or knowledge is stored in computers, and then the scientist analyzes this data using data management, statistical, mathematical, or algorithmic approaches. There's not one tool set that a data scientist uses. And I think somewhat exciting about this is who would have ever thought, I guess, in the spirit of the revenge of the nerds, that at least according to Harvard Business Review, data scientist is the sexiest job of the 21st century. Well, our group at ASU has been very interested in now bringing these data science approaches to large-scale genetics and genomics. And in this work, we're creating a new data science instrument, what we call the next generation cyber capability, or the NGCC. What we're doing is following in the tradition of data gathering science. We're building a scientific instrument for the data science universe, not unlike the large-scale data gathering instruments that we're familiar with in biology, genome sequencer, but in physics, the Hadron Large Collider, in astronomy, the either space telescopes or the Mars lander. So what we're interested in doing is building an instrument that facilitates the work of data science. In data science, we see this instrument as being an integral or elemental whole of three dimensions. One is physical capacity, ultra-high bandwidth networks, large-scale storage measured in hundreds of petabytes to hundreds of terabytes to hundreds of petabytes, and multiple flavors of computation that include high-performance computing, new big data, physical infrastructures that configure the instrumentation in unique ways, as well as rich transactional and large data reservoirs that facilitate the capture and manipulation of excuse me. These physical capacities are complemented by logical capabilities, software, the management of unique software necessary to leverage these physical capacities and to analyze the data, metadata resources that capture or describe the data that's part of this large-scale data resources, and semantics, understanding of the relationship between and among data and data elements. Lastly, in this, a data science instrument is not an instrument unless you bring in the human resources, the transdisciplinary teams that include folks that know how to run the physical infrastructure, engineers that know how to create and manage the logical capabilities, as well as then analysts that know how to leverage the platform, derive what the physical capacity needs to be, and inform the engineers who are building out those individual capacities. So at ASU, our NGCC data science instrument, shown here is a cartoon of this, where we have the logical capabilities that manage context, that perform analysis, that manage the data, and perform transactions, supported with different flavors of high-performance computing and big data computing, managed through transactional resources that have a huge data reservoir that allow us to have large capacity data of what we can see then on the very right side, heterogeneous content composed of many, many different types of data. So we believe the key of this is connectedness, that having the data accessible and having the metadata and semantic infrastructure allows us to ask and answer questions that one simply couldn't do in a different setting. So what I want to close the talk with is a couple examples of how we are leveraging this type of infrastructure today. Again, the future already being here, not a solved problem by any means, but at least the first early inklings that by bringing together this multidimensional data, we can discover things that we couldn't do before, and in fact drive and support evidence generation and understanding of very complex biomedical problems. So a couple examples of where we're using the NGCC as an evidence engine, using raw data resources as clinical reference standards is the starting point here. So in this instance, having these complete collection of resources, we use an infrastructure that we created while I was at the National Cancer Institute called CA Integrator. CA Integrator brings together diverse information on radiology, molecular characterization, as well as deep information on clinical resources. And what I'm going to show is just one example of by having those resources at hand and characterized and defined in a metadata-driven, semantically rich way, we can ask and answer questions in real time. So one way we can ask and answer questions is starting with a particular disease case. In this instance, it would be somebody with GBM, where a individual radiologist has characterized the characteristics of this particular individual's disease. And then by using those characteristics, we can query this big data integrated resource and find or make predictions as to whether someone with this individual's characteristics has differential survival from an individual who, for the vast majority of the population that doesn't have these characteristics. So if you look on this slide, you can see the Kaplan-Meier plot calculated for other people in our deep data resource. What's their survival look like for the, in this instance, 5 individuals that have the same characteristics as this individual, and how does that contrast to the survival of individuals who have different characteristics? Does this make your survival better or worse for glioblastoma multiforme than other types of characterization? We've also actually done this big data technology proof of concept in partnership with our colleagues at Buetow and Hamilton with rich collections of molecular data, data from the Cancer Genome Atlas, data from the NCI Nature Pathway Interaction Database, from the NCI Cancer Gene Index, and the SOPHIC Gene/Drug Database that allows us to ask multiple complicated queries. And in this instance, we can ask and answer queries of the type shown here. So for instance, there are sample clinical research queries that we can say for tumor samples, which are from patients who had progressive disease, we can bring up all of the genes which are in a high amplification chromosomal region and their gene name. So we can actually see what are the genes that might be driving progressive disease based on their amplification status. Similarly, we can actually retrieve the gene expression scores of all the genes that share a high amplification region with a known high expression probe, along with the information about the sex, chemotherapy treatment, and the lifespan. So in other words, by looking at the genes, sort of the converse of the previous experiment, we can retrieve the genes that appear to be influencing one's chemotherapy, that are modified by chemotherapy treatments and have an effect on the overall outcome. Lastly, in this particular set of candidate queries that we can support through this, we can retrieve the set of genes which have a documented association with one or more drugs and that are expressed at or over a specified threshold but are not known in a high amplification region. So we can actually look for genes that aren't being driven by molecular alterations associated with amplifications, but instead by alterations that are as yet uncharacterized. So we could go back and try to figure out what's driving those particular types of modifications. Of course, our big goal is not to be able to ask these clinical research questions, but to really support the transition into the clinic itself. That would allow us to say, given the clinical, demographic, and lifestyle characteristics of the patients, how does survival differ among alternative interventions? What are the adverse events associated with alternative interventions? And what are the quality of life differences associated with alternative interventions? And then based on given all the above, can we make a recommendation from using these as supporting tools? Can we make a recommendation to a physician and or to a patient what might be the alternative courses, or at least inform the patient and or care provider what would be the consequences of alternative intervention in an evidence-based fashion, leveraging all of this heterogeneous and complex data that would be in the system. So I'm going to close with a quick summary. We think that unique insights can come from addressing cancer as a complex adaptive system, and they may help address some of the challenges that we currently face, in particular in cancer. We think it's technically feasible to create and deploy these. We've shown a couple of examples, both in our own house as well as in the community, where this is already happening. We think we get unique coherence by using network models to augment the genome, the sort of linear organization or gene-specific characterization of particular work. And that we believe that, in order to solve these problems, we're going to need to not only have multidimensional data, but actually multidimensional communities to help address these. So I welcome you all. We would love to have you be part of our community. Anyone who would be interested in playing with us and would be interested in collaborating, please feel free to contact me at Kenneth Buetow at asu.edu. And with that, what I wanted to do is point you all to where you need to go. I'll remind you of this actually after I open up the session for questions, but to point you to this URL, www.bioconferencelive.com, in order to go get the CME or CE credits associated with this particular module. That's where you would go to actually perform the next steps to get the CME CE credit. So with that, with a few minutes left, I'm going to open the floor for questions. I'm going to see if we actually have any questions that have been put forward. Again, feel free to type these into your particular viewing screen. As of right now, I don't have any formal questions, but we'll wait a minute or two and see if folks have any specific questions. Again, while we're waiting, I'll remind you for capture of the continuing education or continuing medical education, you can go to www.bioconferencelive.com. All right, well, at this point, I don't see any questions. I'm happy to entertain them if there are others that are interested, but in the absence thereof, we'll wait for just a minute. So feel free, if you have questions, to enter them into the QA box. So I guess I'll take, at this point, seeing no specific questions, I want to thank you all for your attention and participation. Oh, okay, I just got a question. So the question is before me, what are some of the exposome characteristics that you've derived? So in this instance, we're actually not deriving exposome characteristics, we're actually capturing them. So in this instance, they would be quite commonly exposures. So for instance, in work that I'm working on that I didn't present in today's presentation, some of the exposome work I'm studying is in liver cancer. It would include the state of whether you are infected by one or more of the hepatitis viruses, hepatitis B, hepatitis C. It could also include whether your obesity status. So we're actually very interested in looking at how one's obesity status drives development of liver disease and or liver cancer. Other questions? So it says, do you work on, the next question is, do you work on ODE or PDE simulations? So for the most part right now, we haven't been doing simulations tied to the particular work, although the infrastructure that we have actually would support any of these different types of flavors of simulations. Other questions? The next question is in 1971. President Richard Nixon announced a war on cancer. How long do we need to wait for real treatments? I actually think that's an appropriate and fair question. I think part of the challenge that we faced in the cancer community and part of, I think, the new opportunity before us is that we spent a lot of time taking cancer apart, understanding its individual components through reductionist paradigms. I think the opportunity that's before us right now is to synthesize or integrate those pieces. So I'm cautiously optimistic that as we start the kind of work that I just described in this talk of not looking or hoping that there's going to be a single magic bullet drug for a single gene, but recognize that, as we saw for GBM, there's multiple networks that have multiple alterations, that if we start to assemble those pieces in comprehensive strategies, that we're going to have much more effective treatments. Now, I'd like to think that that's going to happen very, very quickly. But that said, I think that that transition is occurring as we speak. And you'll look, as we see right now, from calls from the NIH, for instance, for its big data to knowledge transformations, we can see that, in fact, there is real work going on right now to try to change the characteristics of those. So I would like to say that the breakthrough changes are just around the corner, but I actually want to not generate the ongoing hype that we've had in the cancer community for a long time. And I think we still have a bit of a slog ahead of us, but at a recent conference that I participated in, there's significant hope that within the next decade, many of the major cancers that we face today that are life-threatening will be transformed into not life-threatening but chronic manageable diseases. The next question asks if am I looking at vitamin D status. In the specific work that I'm working on right now, I'm not working particularly on vitamin D status, but I know for a variety of different cancers, vitamin D status is a significant interest. And in theory, in the exposome space would be one of the critical things I think we would want to be able to bring in to the types of analysis we perform. Another question says, do you foresee a pushback in big data usage in life sciences after the NSA surveillance leakage? The short answer is, I hope not, partially because unlike the NSA, we in biomedicine have been very, very upfront with use and reuse of data. We, unlike the NSA, consent and or engage the patients about what the data is and upfront tell them what will be the circumstances, and they have the right to determine when their data will be used and won't be used. That said, I think we are going to see changes in how liberal or comfortable people are. So I think that there's likely to be, some pushback. Okay. The next question says that my big data concerns fall well within the realm of estimation theory and decision theory. You mentioned effectively through, let's see, I think this is what drugs, so I'm not sure I can actually formally answer what drugs have changed. Let's see, from this I don't think I have the whole question, so I said, the next question I see is, what drugs have changed pancreatic cancer prognosis and any specific gene markers? I'm not certainly an expert in that space, so I unfortunately will have to defer that to someone else, so my apologies for not being able to help with that. Any other questions that we have? So our next question is, how complex can be the complexity of cancer if epigenetic complexes enhance the complexity of cancer treatment? I think that it actually can be quite complex. The epigenetic changes actually have profound ways to change large-scale components. Now, that said, it's through the embracing of complexity that we can actually see those changes. And if we actually look on a gene-by-gene basis, the signals look chaotic. But if we embrace the complexity, what we see, in fact, is again, a coherence of regions that are being turned on and or turned off by epigenetic changes, and that these epigenetic cancer treatments actually then may actually facilitate the transformation or deactivation of cancer and oncogenic phenotypes. Another question is, how will big data impact the role of subject matter expertise in the modern world? I think it's actually, we're in the spirit of, I opened this talk with both revolution and renaissance, I actually think it's going to be revolutionary. I think by releasing the data and harvesting this diverse resources that historically have been locked away and inaccessible, subject matter expertise will be greatly expanded. So in other words, one won't have to master every domain in order to utilize all of the domains of data and knowledge. So much the same as the web allows me to ask and answer questions in real time to receive information I would have never known about, it changes that. Big data will facilitate that as well. And then I think for last question is it necessary to work with computational studies that should have mathematical expertise? I think the cool part of the new big data problem is that, in fact, it doesn't require just a simple single approach to the problem. Okay? I think we're out of time at this point. I want to thank you all for your participation. I think it's been, I've enjoyed having the opportunity to interact with you. Thank you for your questions. And at this point, I thank you for your participation and say goodbye. So thank you.